Bio Enzymes Part III will focus on enzyme inhibitors and metabolic pathways. The essential idea is metabolic pathways are regulated in response to the cell's needs. Here is the outline of topics for the 3 parts of the enzyme unit. You can use this slide to find your way to the section you need. This movie is focused here. Here's the IB syllabus statement that kicks off this movie. Explain the difference between competitive and non-competitive inhibition. Provide one example of each. Distinguish different types of inhibition from graphs at specified substrate concentration. Enzymes are inhibited by 2 broad classes of inhibitors, the competitive and non-competitive inhibitors. But be careful here. The inhibition of enzyme activity has some distinct advantages in the cell, or in other words, inhibition as a term should not be interpreted as a necessarily negative event. Pay close attention as we move forward. Now, the 2 broad classes of inhibitors are competitive inhibitors. The active site is directly involved with the inhibitor. There is reversible binding of the inhibitor to the active site or irreversible binding, and this is poisonous. In non-competitive inhibitors, an other site, an allosteric site, is involved along with the active site. The inhibitor reversibly binds to the other site, and non-competitive inhibition is the central mechanism of metabolic feedback inhibition. Here is a diagram that explains competitive inhibition. On the left, we can see the active site available to the substrate. On the right, we can see that the active site is blocked by a molecule known as an inhibitor. The inhibitor prevents the substrate from entering the active site. Now, the term competitive is used to describe the inhibition here because the substrate must compete with the inhibitor for the active site. Now, this inhibitor could bind to the active site in a loose fashion, a reversible binding, or the inhibitor could bind permanently, that's irreversibly. If the inhibitor binds irreversibly, then the enzyme is permanently disabled. Poisons are often irreversible competitive inhibitors. Here's another diagram displaying competitive inhibition. The substrate and the inhibitor both compete for the active site. Examples of competitive inhibitors are sarin gas on an enzyme known as acetylcholinesterase and antibiotics. Competitive inhibitors are structurally similar to the substrate. In this example, we have succinate, which is metabolically transformed into fumarate by the enzyme succinate dehydrogenase. But malonate is a competitive inhibitor for the enzyme succinate dehydrogenase. You can see the structural similarity of malonate with the normal substrate of the enzyme, succinate. Evidence of competitive inhibition can be seen when examining reaction rate versus substrate concentration. In the absence of an inhibitor, we have the blue curve, and in the presence of the inhibitor, we have the green curve. At low substrate concentrations, the inhibitor lowers the rate of reaction substantially. But as substrate concentrations increase, more and more substrate would allow the substrate to outcompete the inhibitor for the active site of the enzyme. At high concentrations of substrate, the rate of the reaction is the same as if the inhibitor were not even present. In other words, so much substrate is available to the enzyme that the substrate outcompetes the inhibitor for the active site, allowing the reaction rate to reach a maximum. This diagram displays non-competitive inhibition. In non-competitive inhibition, a molecule binds to a site other than the active site to cause a conformational shape change in the active site of the enzyme, altering the shape such that it cannot bind the substrate. The reaction is inhibited. The other site to which the inhibitor binds is called the allosteric site. Allo means other. Here's another diagram of non-competitive inhibition. The inhibitor binds to the allosteric site, causing a conformational shape change in the entire enzyme. that alters the shape of the active site. The substrate cannot bind to the active site, so the reaction is inhibited. Evidence of non-competitive inhibition can be seen when examining reaction rate versus substrate concentration. We have the normal reaction curve here in blue, and then the reaction with the inhibitor here in green. You can see that the inhibitor inhibits the reaction at all substrate concentrations. In other words, with the inhibitor, the rate of reaction is lower at all substrate concentrations. Contrast this with competitive inhibition. Review competitive inhibition as need be. The fact that the inhibitor is not competing for the active site with the substrate means that no matter how much substrate is present, the reaction will always display some inhibition, as can be seen here. Here's a quick review. Competitive inhibitors bind to the active site, competing with the substrate for the active site. Competitive inhibitors can bind to the active site reversibly or irreversibly. Irreversible inhibition is the mechanism of poisons. And here is a review of non-competitive inhibition. The inhibitor binds to an allosteric site, altering the shape of the enzyme, which alters the active site. The substrate cannot bind, and the reaction is inhibited. And here's a review of reaction rate versus substrate concentration in the absence of inhibitors and the presence of inhibitors, the competitive inhibitor curve, and the non-competitive inhibition curve. When substrate concentrations are high, the substrate can outcompete the competitive inhibitor, so reaction rates reach a maximum. But non-competitive inhibitors are not competing with the substrate for the active site, so the rate of reaction in the non-competitive inhibitor curve is inhibited at any substrate concentration. Study this slide. Again, here's a comparison of competitive inhibition, where the inhibitor competes with the substrate for the active site, and non-competitive inhibition, where the inhibitor binds to an allosteric site, causing conformational shape change in the enzyme that changes the shape of the active site such that it cannot bind the substrate, inhibiting the reaction. Study this slide. So with non-competitive inhibition in your control, let's expand on non-competitive inhibition as a mechanism for metabolic control through negative feedback. Now imagine if the enzyme catalyzes the first reaction in a series of reactions that ultimately produces a product, and the product could, upon an increase in concentration, bind to the first enzyme at an allosteric site. The reaction would be inhibited. This would slow the production of the product. As the production of the product slowed, the inhibition would be released, and the rate of reaction would return to normal, allowing more product to be produced. And as the product was made, it would in turn bind to the first enzyme at an allosteric site to slow the reaction, responsible for its own making. This is an on-off switch, negative feedback control of metabolism. Pay close attention. Here are the two IB syllabus statements of importance. Explain how metabolic pathways can be controlled by end product inhibition. State that metabolic pathways consist of chains and cycles of enzyme-catalyzed reactions. Metabolic pathways can be chains through a series of intermediates to produce a product, or cycles where the molecule at the start is regenerated through a series of intermediates. So let's look closely at how metabolic pathways can be controlled by non-competitive end product inhibition. A metabolic pathway is shown here, starting with a molecule that is enzyme catalyzed through a series of intermediates leading to product D. Now imagine that product D was a non-competitive inhibitor of enzyme 1. As product D accumulates, it would serve to inhibit the metabolic pathway, slowing its own production. As the concentration of product D declined, the inhibition of enzyme 1 would be released, and the metabolic pathway would operate to produce product D, and so on. So here's an example. the amino acid threonine is metabolized to amino acid isoleucine through a series of metabolic steps. Isoleucine, in turn, is an end product that inhibits the first enzyme in the pathway, shutting off its own production until isoleucine concentrations decrease, allowing the enzyme to function without inhibition, so the production of isoleucine resumes, and so on. Metabolic pathways can be controlled by end product non-competitive inhibition. This diagram displays the threonine to isoleucine example again. Isoleucine acts as a non-competitive inhibitor on the first enzyme of the pathway known as threonine deaminase. End product inhibition provides homeostatic control of metabolism through negative feedback. An increase in isoleucine results in a decrease through negative feedback control of isoleucine. However, a decrease in isoleucine frees the enzyme up of the inhibition that would serve to increase the production of isoleucine, etc., etc. Here is the full IB syllabus statement. Explain the role of allostery in the control of metabolic pathways by end product inhibition. Use the example of the conversion of threonine into isoleucine. Here's another image of the threonine to isoleucine example. Study this image as need be. The end product becomes a non-competitive inhibitor of threonine deaminase by binding to the allosteric site and causing a conformational shape change in the enzyme so that the active site no longer binds the substrate threonine. When that shuts down the pathway that leads to isoleucine, the concentrations of isoleucine decline. This in turn releases the inhibition on the first enzyme, which causes the pathway to resume, causing a further production of isoleucine. As a note, bioinformatic databases allow scientists to find molecules that might inhibit enzymes in disease-producing organisms. Malaria, for example, has become resistant to many drugs. Databases need to be used to find inhibitors of the malarial enzymes. Here is a respiration pathway that produces ATP starting from glucose. Once enough ATP is formed, ATP feeds back to inhibit an enzyme at work early in the pathway. That enzyme is called phosphofructokinase, or PFK for short. Interestingly, when ATP is spent and becomes low in concentration, ADP, which is increased, causes PFK to act more rapidly. In other words, ADP binds to an allosteric site on PFK to cause the active site of PFK to more readily bind the substrate, fructose 6-phosphate. Now, as I come to the end of this movie, let's step away from enzymes briefly, but talk about molecules that display allostery, that have multiple binding sites. These molecules do have multiple subunits, multiple binding sites. They exist in equilibrium between an active and an inactive form, and they display what's called cooperativity, as they're always in motion as a result of conformational shape change. For example, in this diagram, the inactive shape, inactive form here is on the left, but as a molecule binds to one subunit, the entire structure undergoes conformational shape change to more readily bind to other molecules. The subunits open progressively more and more as molecules bind. Now follow the story here. Hemoglobin is just such a molecule. Hemoglobin is composed of 4 polypeptide chains, each with a heme group that binds oxygen. In other words, each hemoglobin molecule binds 4 oxygen molecules. Stay tuned. So as hemoglobin binds oxygen, hemoglobin takes on one conformational shape, and as hemoglobin releases it, it takes on another. So as hemoglobin is moved through the circulatory system from body to lungs and back, it's rocking back and forth between 2 conformational states. Allosteric molecules with multiple binding sites, like hemoglobin, display S-shaped curves of rate of reaction and substrate concentration. Within a very narrow range of substrate concentration, the reaction rate increases dramatically. As one substrate binds, the molecule undergoes conformational shape change, allowing more to bind, and the reaction rate increases rapidly with a small change in the substrate concentration. We can see the S-shaped curve when we examine hemoglobin's oxygen saturation versus concentration of oxygen. The y-axis is saturation. You could almost think of this as rate of reaction, and oxygen is like the substrate. Now be careful. Hemoglobin is not an enzyme. But in any case, the steep increase in saturation with oxygen through this fairly narrow range of oxygen concentrations results from hemoglobin's cooperativity. As one oxygen molecule binds, hemoglobin undergoes conformational shape change that facilitates more oxygen binding. As hemoglobin binds and releases oxygen through its course in the circulatory system, hemoglobin's shape rocks back and forth between the oxygenated and deoxygenated conditions. And that brings us to the end of IB Bio Enzymes Part 3.